Okay, thank you very much, Kim, and thank you all for joining. It's a very timely, I guess, topic to be discussing. So, how can biochemical engineer harness biology to address global challenges? So, we'll be talking a bit about biochemical engineering in general in the beginning, just as an introduction for those of you that might not be 100% fully aware of what it is, or maybe some of the details of what it entails, and then we'll move on to actually talking about UCL and life in London a bit in general, the facilities over here, but the research areas of the department, and then the structure of the MSc itself. So it should give you a nice idea, a nice overview of what life will be over the year-long MSc course. So let's start, as a warm-up, sort of couple of slides on what is biochemical engineering. Now, biochemical engineers, let's say that if there was such thing as a textbook definition, that would be biochemical engineers apply the principles of biology, chemistry, and engineering to produce useful products such as biopharmaceuticals, biofuels, biopolymers, and industrial enzymes. And that's a bit broad. So, sometimes I like to draw some analogies to maybe put things a bit into perspective. So, um, much like any other engineering discipline, so all engineering disciplines are based on addressing a need of our society. Okay, so we have various engineering disciplines. So I'll have sets of pictures here that will correspond to various engineering disciplines, and I'll be asking you some questions. If you could type your answers in chat, that would be great. So in this particular case, what is the need that is depicted in this picture? And in case it's not clear through the web link on your screens, this is a picture of a hut. So what is the need that's being represented by this picture? If you could type your answers in chat, that would be great. Any ideas? What a place to live home? Yes, exactly, Justine. So it's the need for shelter, right? We, as human beings, we need to have a roof over our head, so to speak, and therefore we undergo a design process where we say this is where the wall will be, this is how big, how long, how tall the wall will be, this is where will be the window, the wall, the door, and so on and so forth. We have a manufacturing step, and then we have the final product, which will be, either a house or a high rise, or depending on what we're building. So obviously, this first set of 4 pictures depicts civil or structural engineering, right? So the engineers that build our houses and homes and blocks of flats and so on. And similarly, can anyone guess what this second set of pictures, which engineering discipline is described by the second set of pictures? Can you type your answer in chat, please? Exactly, Sharon. Yeah. So it's mechanical engineering, right? So we have the need for transportation. Again, we undergo a design process. We have a manufacturing step where we assemble all the cogs and bolts and what have you together. And then we have the final product, which will be some form of transportation. Now, Uh, moving on, onto biochemical engineering, we have the need, or one of the needs that biochemical engineering is trying to address is the need to maintain our health and well-being, right? So unfortunately, as human beings, we do get sick, and we need to produce medicine in order to heal and get better. Similar to every other engineering discipline, we have a design process. Now, what is unique about the design process in biochemical engineering, and this is a trait shared with chemical engineering because the two disciplines are fairly close together, is that the nature of the materials that we're handling in the design process changes, right? So if you think about it, if I start my fermentation, I will start with a small number of cells, and I will have certain properties in my fermentation broth, or the liquid, let's say, inside my reactor, not to use technical terms. And as time progresses and my cell populated become more and more, they start consuming the nutrients in there, they start producing whatever it is we're trying to produce. So the nature of that composition in that liquid changes over time. And that happens throughout the entire process, right? When I'm purifying my process, I'm changing the concentration, and so on and so forth. So this dynamic nature, this ever-changing nature of the material that is being processed is unique to the process engineering disciplines, which is essentially bioprocess and chemical, biochemical and chemical engineering. Just to draw a contrast here, a brick over any perceivable amount of time will not drastically change, right? Definitely not in the manufacturing process in and of itself, right? Maybe over 50, 100 years, a brick will change slightly in composition, but not during the manufacturing step itself. Um, we also have a manufacturing step, much like all other engineering disciplines. Um, and we have the final product, which in this case would be some sort of pharmaceutical drug, and it could be a orally administered pill or a vaccine or what have you, depending on the substance and the nature of the drug itself. So uh, building up on that topic of, let's say, biopharmaceuticals, what is it that a biochemical engineer is tasked to do? Okay, so let's think of how, and I think it's a very timely period to be having this discussion. People, you're obviously listening to the news with what's happening with the coronavirus outbreak, and you hear or you read on the news that people are trying to find a vaccine or an antibody drug. So how is that? Where would the biochemical engineer be involved in this process? So what happens is biologists, pharmacists, genetic engineers, depending on the variety of disciplines that would be involved, are trying to figure out the structure of either a vaccine or an antibody that would be effective against a particular disease target. Okay, so during this experimentation, where they're trying to discover this new pharmaceutical substance, they would be operating usually in something equivalent to a petri dish, right? Very frequently an actual petri dish. And for those of you that don't know, a petri dish is the small plastic cup. It's very shallow plastic cup, let's say, that's roughly up to 3 milliliters in volume, so quite small volumes. It's static, therefore there's no agitation. It's not being shaken around. It just sits on a table or a bench. And the cell numbers, if we're growing cells in the petri dish, are generally quite low. Too, you don't get a huge amount of cells. And the reason for that is because we're growing cells in suboptimal conditions. But that's where discovery happens. So, when you hear, hopefully, in a couple of weeks' time at most, this is me being optimistic, that the University of Oxford or whatever company has discovered a new vaccine or a new antibody that's effective against the coronavirus, it will be at a similar scale as the one shown here. Now, what happens once this is discovered is where biochemical engineers start getting involved. So we have to take now this process and this cell and this product that has been produced in the petri dish and replicate it in a scale big enough to satisfy global demand. And seeing the number of patients worldwide being in the hundreds of thousands for the coronavirus, you can imagine how much of this new drug we would need to produce. So what biochemical engineers do is they take the process and the product produced at the petri dish or at the discovery scale, just to be a bit more general, and we do what is known as a scale up. We try to bring it to start growing and repeating and replicating this process that produces the exact same drug with the exact same quality attributes at larger and larger scales. So we will start with what we call a lab or a pilot scale, which will be between 50 milliliters up to 5 liters. And eventually we'll want to, and these will be static, shaken, or stirred. We will have some limited control on the environment, so we'll be able to control temperature, maybe carbon dioxide, and we can perform some preliminary process optimization, like what would the media composition be like, how much should we need to feed it, what's the optimal temperature, stuff like that. And eventually we want to lead up to the industrial scale, where we're talking anything between 20 to 10,000 liters. There we have full control over the environment. We can stir, we can mix the culture continuously. We can provide air, we can provide carbon dioxide. we have full control more or less over the environment. But we have other challenges due to the magnitude of the scale at which we're operating. So, you know, if you think about a 20,000 liter reactor, think about it, you know, if it's something that's maybe a meter in diameter or a meter and a half in diameter, it would be in height almost up to the second story of a block of flats, right? So that's quite tall. So you can imagine now cells that are in the bottom of the reactor are compacted, are being crushed from the weight of all the liquid that's above them. So you start having some other challenges. But nevertheless, it is the job of the biochemical engineer starts from here, from the 3 milliliter scale, and needs to design equipment and processes and conditions that will ensure that whatever product and at the same efficiency and quality characteristics was produced at this scale can be produced in a financially sustainable way at the 20 or 10,000 liter scale. Sorry, I'm going to just, I see chats popping up, so I'm just going to make sure that you can hear me. So I'm just going to bring up my chat window. That's one of the things I will be addressing. So, right, so that's what biochemical engineers have to do. And imagine that the example that we just talked through only refers to the fermentation part of the process, right? Where we grow cells and the cells produce the pharmaceutical product. But actually, that's a very small part of the overall procedure that needs to be followed in order to have a vial or a tablet on the shelf that a patient can take. So once the fermentation has finished, there is a number of purification and clarification and formulation steps where we separate the cells and, or let's say, the unwanted substances from our drug substance all the way until we formulate a pill or a vaccine or whatever its final pharmaceutical form needs to be. So you can imagine that we need to repeat all of these steps for each and every step all the way until we end up with a pill or the final pharmaceutical form. And therefore, it really comes as no surprise that the average timeline for the discovery of a new drug is roughly between 7 and 10 years, right? This takes into account the efforts in actually identifying the drug. So what biochemical engineers are trying to do is we're trying to shorten the timeline as much as possible. So if we can bring this 7 to 10 years down to, let's say, 3 to 5 years, then that would be a massive improvement. The reason for that is approximately according to statistics, only 10% of the molecules discovered at the lab scale as successful candidates against a particular disease ever make it to the shelves, right? So the cost of the 90% of pharmaceutics that have failed, so all that R&D development costs, the media, consumable costs, staff costs, and so on, all of that is baked into the 10% of drugs that are actually successful and on the market, because, you know, big pharmas or any pharma company needs to be financially sustainable, so they need to mitigate those costs. So by reducing the timeline even by 10 or 20%, you would be reducing effectively the cost of medicine towards the patient and national health systems. So in essence, that is what biochemical engineers try to do. And the kind of questions that we need to answer sort of on a daily basis is, how will I manufacture it? How will we make this drug? How much can we make? What's the maximum productivity, or what should be the productivity? Should we operate at 100 liters, at 1,000 liters, 10,000 liters? Can we make sure that whatever process we design meets legal regulatory requirements? Just to give you an idea, for antibody products that are meant to be administered to humans, the purity in the final formulation must be above 99.9%. So that requires a very refined, very sophisticated way of clarifying and removing all impurities. How much will it cost? Can we make it cost effective? And do we have a commercially, financially sustainable process? So it's a multidisciplinary field by nature. It involves a lot of engineering, which, you know, is physics and maths. It involves a lot of biology and biochemistry. But it also involves a lot of business decisions on, particularly around patenting and making sure you have commercially or financially sustainable processes. Now, what are some of the global challenges that biochemical engineers have contributed towards? And again, this is a slide I routinely have, but I guess that today is more relevant than other times that I'm giving this presentation. So it's the response to epidemics and pandemics like the one we're living through now, unfortunately. So the idea is, and why people think that we can have a product on the shelves anywhere between 12 to 18 months from now, as opposed to the usual, 5 to 7 to 10 years, is and that's the most significant process, is from a legal and regulatory point of view, this will be fast tracked. So everything will happen much quicker, whereas usually this is a quite slow and lengthy process. But also over time, biochemical engineers have really refined the concept of platform processes. So essentially what we have is even without knowing the product, right? We don't know what the final form of the antibody will be or what the final form of the vaccine will be. We already know what cells we're going to use to produce this product, right? Because we've slowly moved towards this concept of a platform process. So if it needs a mammalian host expression system, it will most likely be either a CHO, a Chinese hamster ovary cell, or a HEC cell. If it's a bacterial-based production, it will most likely be an E. coli-based production, right? So we know we already have the cells ready and waiting for the molecular biologist and the geneticist to give us the final DNA sequence of the drug target, and then we can just plug it into our cells, and we already have a lot of knowledge about how that process should look like. Now, on top of that, biochemical engineers work a lot on process intensification and continuous processing. That means that we can produce essentially more using a smaller volume of material. This is very appealing, A, because it's cost effective, but also B, because it allows us to set up new capacity very, very quickly. It's much easier to set up a facility around a 1,000-liter fermenter than it is to set up a facility around the 10,000-liter fermenter. And what helps even more in that direction is the development of single-use technologies. So, in my slides, in a couple of slides ago, I showed you this big industrial fermenter that was made out of stainless steel and required quite substantial infrastructure to hold the thing in place. So what biochemical engineers have developed over the past maybe 10, 15 years is what we now call single-use technologies, where instead of using this built infrastructure, these stainless steel reactors, we use single-use reactors, which there's no way of describing them other than a very sophisticated plastic bag. But essentially, imagine a reactor made out of plastic with all the components that a normal reactor would have, but everything made out of plastic and everything pre-assembled. So you can very easily set up a process using these single-use technologies and start producing whatever it is you need to be producing. I've already touched upon the need to have affordable medicine, and one of the things that we're trying to do here as biochemical engineers is reduce the time to market. We use several tools. We use the concept of quality by design. So we use mathematical analysis and modeling techniques to design more efficient and better performing processes. But now, in the past 5 years, definitely, and efforts are ramping up, we rely a lot on robotics and artificial intelligence to be able to screen even more drug candidates and even more process conditions and derive better processing and more efficient processing conditions more quickly. So along these two challenges, some of the products that you could be associated with as a biochemical engineer would be vaccines, antibodies, and recently cell and gene therapies. But drugs are and pharma is one side of the equation. Biochemical engineers are involved in anything that has to do with the processing of cells or their components. So we also contribute quite a bit towards the energy shortage that we're experiencing in the sense of fossil fuels are limited, and we need to come up with alternate forms of fuel that are sustainable and environmentally friendly. So biochemical engineers have worked extensively on the concept of biorefineries, where you create biofuels, but also use essentially as much of the cellular biomass as you possibly can to extract other valuable components. So essentially nothing to waste kind of mentality. And another area where we're actively involved with is something called bioremediation. So bioremediation is, let's imagine you have a body of water that's contaminated. This could be an overgrown lake, or this could be a river flowing very close to a mine that's been contaminated through the actions of the mining industry, so on. So it has toxic compounds that are very difficult to otherwise process, and we need to process huge volumes of it. So biochemical engineers have developed a series of either naturally occurring microorganisms or genetically modified microorganisms that are able to essentially be dispersed in the polluted body of water and eat up the contaminants. And that's a process known as bioremediation, where we use cells to essentially clean up or remove toxic substances from either contaminated soil or water. So before I move on, I'd like to address the question that has been raised on what is UCL Biochemical Engineering's involvement in the coronavirus crisis. So one of the areas of active research in the department is on vaccine technology, and we have two very big grants that essentially are led by the department. One is funded by the Bill and Melinda Gates Foundation, and the objective of that sort of research consortium is to reduce the cost of vaccines to, I believe, roughly $2 a dose. And we have, a couple of years ago, we were also awarded another big grant that we call the VAX Hub. So that a hub means this is again a consortium of many universities. So it's UCL Biochemical Engineering, it's Imperial College, and it's the University of Oxford that are all working under this umbrella of the VAX Hub. And actually, when you read the news that Oxford is trying to develop, the University of Oxford is trying to develop a drug and is hoping to go into clinical trials soon, a lot of the funding of the VAX Hub has been diverted towards those efforts. And the idea is that once the drug target is successful and has been identified, UCL Biochemical Engineering could be involved in designing the process and scaling up the process and so on. In addition, we've been approached by a couple of companies that produce antibody-based detection tests for the coronavirus, asking us for either expertise and advice, or whether they could use our facilities to do some small production runs. It's things are moving forward at a quite rapid pace, and we try to contribute as best we can to efforts around the world. And to be honest, this is not effort that any single company or university could claim as their own. I think definitely in the UK, all of academia and even a lot of normally competing industries have come together in trying to figure out a solution to this. But so, UCL Biochemical Engineering is doing its part as best it can. So now a bit about studying at UCL. So for those of you that are not from London, I'm highlighting here in the orange box where UCL is located. So this is the main campus of UCL. It actually spans quite a larger area around that box, but let's say the heart of the campus is within that orange box. And and if I bring up a couple of the notable sites around London, you will see that UCL is quite centrally located. So we have the British Museum, we have King's Cross, we have the Big Ben and the London Eye. So Madame Tussauds. So everything is very, very close to UCL. So UCL is really in the heart of London. In terms of its history, UCL was founded in 1826, and it was founded on the principle of equal admission irrespective of class, sex, race, or religion. So it was the first university to offer. So basically, back then, if you wanted to study in university, you had to be white, male, upper class. Right? That was your only option. If you didn't tick all those three boxes, you couldn't study. At least not in the UK. And UCL was the first one to break that barrier. Uh, and this ethos of acceptance and welcoming everyone, irrespective of background or any other possible uh, way people unfortunately find to discriminate themselves, still stands true today. Nobel prizes have been awarded to 29 people who were either researchers or alumni or previous students of UCL. And today, UCL is consistently within the top 10 universities worldwide. It boasts more than 4,000 academic staff in 72 departments. So it's an all-encompassing university. It's not specialized in any particular area. It has literally departments on everything you can imagine: arts, sciences, law, engineering, medicine, everything. Um, we have a fairly equal ratio between men and women, so it's around 52% women, 48% men, and we have students from over 150 countries. So our motto is London's Global University. Now, one of the benefits of joining such a big university is that you get access to a lot of centralized facilities that you might not necessarily find in smaller places. And just to go over a couple of examples, we have a very well-equipped library that's open 24/7 to all students. We recently, as in just barely more than a year ago, opened up our new student center. So this is a 5 or 6 story building right next to the department. It's literally adjacent to the department. That's dedicated to students. So that has recreational areas, student study spaces, quiet places, meditation places, group workplaces. It's really a building dedicated to students. And it's actually has been very well received by the community so far. We have computer cluster rooms spread throughout campus. So we have 14 of them where you can go and work on a bigger screen, if you don't want to be working off your laptop all the time. We have a language center that you can sign up if you want to improve your English or if you want to learn another foreign language. We have museums and collections that are open to the public as well as to students on campus. And we have student um, clubs and societies for anything you can imagine: sport, theater, politics, anything you can imagine there is a society for. Right next to the student center, so 2 buildings away from the Department of Biochemical Engineering, we have the Bloomsbury Theatre. So that's a full-fledged West End theatre. And on top of that sits the gym. It's called the Bloomsbury Fitness Center, which is available for students who want to work out and keep fit. On top of that, we have a dedicated department for career support. And down here, you will see the links that you can visit to learn more about each of the facilities that I'm talking about. So careers at UCL can help you with one-to-one appointments where they go over your CV and give you some tips and tricks on how to improve and how to present it depending on the sector that you're applying to. They can have wider career discussions, so general career advice, but they can also book mock interviews. So you'll you'll host an interview, a trial interview, let's say, with someone from careers at UCL that has experience in HR and hiring. And they organize a number of career fairs throughout the year. So these career fairs can be sector specific. So let's say engineering only companies come, or they can be general career fairs where you can have employers or prospective employers from any sector. They also run a number of skill development workshops. Another useful service, particularly if you're not from London, is the UCL Accommodation, and they offer both halls of residence, but also private accommodation. So it's a big help if you're new to London and trying to find a place to live. I would definitely recommend starting from having a look at UCL Accommodation. Um, and we have a dedicated portal that gives sort of day-to-day information that you need to know about how the campus operates and where you can find help, and key dates about the term, but also more practical information like how do I use the tube, how do I open a bank account, how do I pay my bills in the UK, and stuff like that. So this is very useful for students coming from outside the UK. And there's almost a subversion or a subsection of that portal that's dedicated exclusively to new students. So that will have your checklist of what do I need to have with me on my first day in college when I go to enroll, what checks do I need to do, what paperwork should I have with me, what's the order of steps that I should follow. So really guiding you through the enrollment process. We have our student support and well-being that's based normally on the in the student center, and this is an exceptional team of professionals that are there to help you. It's a long and tough year. People will get stressed over exams or over coursework deadline, and living in London on your own, you might feel lonely on a day or two. So student support and well-being is there to support you. It's a very friendly group of professionals that are very passionate about what they do. I've interacted with them quite a bit, and it's always been a very, very pleasant experience. And finally, we have the UCL Union, which is student-led. You will notice that the URL is not from UCL; it's completely independent. And the student union is responsible for all the clubs and societies that are operating in campus, but also for running a lot of the cafeterias that we have on campus. So those are student organized and student run. Okay, so moving on now to the department itself. We're a small and friendly department. I can manage to fit everyone in a slide. And our unofficial motto is that you sort of join the department for life. And the reason for that is because exactly we're a fairly small department. So we're around 25, 26 members of teaching staff. And our cohort, so our intake for the MSc is usually around maybe 40 students, give or take. So it's more than, so it's almost, 1.5 students per academic, right? So you get a very close and very personal relationship with both your peers in your cohort, but also the member of staff. So that leads to an alumni network that's very, very engaged with what's going on in the department, and keeps in touch actively and actually participates in a lot of the activities that we run throughout the year. So the department itself was the first department in the UK to dedicate itself solely to biochemical engineering. So most departments around the world are chemical and biochemical, and it's one of the few departments around the world that's pure biochemical engineering. And in the latest research excellence exercise that's conducted by the UK government every 5 or 6 years, 90% of our staff was classed as either world leading or internationally excellent. But research excellence is not the only thing we strive for. We've received a number of prizes for our teaching practices, including the Queen's Anniversary Prize for translating research and bringing it into our education and into our degrees. On top of that, we're quite active in terms of enterprise. So we now count I should be updating my slide, 4 successful spin-out companies. And when I say successful, I mean They have successfully raised their first capital from investors, and 3 out of those 4 have been bought out by larger companies and are actually producing their product actively. The 4th one is very recent. It was spun out last year, and it has just received its first round of funding successfully. Now, one of the benefits and one of the key, I'd say, features of how the department operates is that we collaborate extensively with industry. This helps both because, as I said, our alumni network is very actively engaged with what goes on in the department, and obviously people that graduate from biochemical engineering go to work for these big biotech companies. But also the nature of our research is actually very, very collaborative with industry itself. So this is not an exhaustive list of the companies that we work with, but this is more a list of companies we very frequently collaborate with. And the way this will benefit you as MSc students is that throughout the year, you'll have the chance to meet and interact with people and representatives from these companies. So for example, would be you could have a poster session for one of the modules, or it could be for your research or design project at the end. And your assessors, the people grading you and giving you feedback, would be senior scientists and senior managers from industry. Some of your modules and some of your lectures will be delivered from people from industry. So you will also be part of the MBI program, which I will talk to you about towards the end of my presentation. But essentially, it's a specialized training program designed for senior scientists in industry that want to retrain in new areas or want to remain up to speed with the latest development in a particular area. And as MSc students, you will be taking some of those MBI modules. So throughout the year, there will be sessions where the person sitting next to you as a student is actually a person from industry. And during the the social breaks, like the coffee breaks and the lunch breaks, you can reach up to them, start building your network, and start getting some advice. How did you apply for that job, or where are they looking for? How should my CV be, and stuff like that. And just to give you an idea of how our research finds its way onto the MSc research projects, this is just a sample of projects taken from the past 4 years. So you can see it ranges from mammalian cell culture to cell and gene therapies to genome editing of E. coli or cell-free production systems, computational fluid dynamics, membrane vesicles for cancer therapy. So it's a very broad area of a broad spectrum of areas that are covered, and that reflects the research that we do in the department as well. So onto the MSc itself. So the MSc has 3 distinct routes that are tailored based on the undergraduate background of the students coming in. So the first route, it's called MSc Biochemical Engineering for Scientists. For all intents and purposes, all the routes are equal. Your final degree will say MSc Biochemical Engineering, irrespective of the route. It will just also have the route indicated beneath that. So the first one for scientists is intended for people with a background in sciences or life sciences. So typical intakes would be chemistry or biochemistry, microbiology, applied and molecular biology, those sorts of things. And that focuses on really taking you as a graduated scientist and retraining you as an engineer. So giving you all the fundamental background on fluid mechanics, reactor design, all the basic engineering concepts that you need to know in order to be a professional biochemical engineer. The route for engineers is suited for anyone with a process engineering background. So this will be chemical or process or manufacturing engineering, depending on where you are around the world. It's called slightly different names. But depending on the nature of the degree, it could include things like mechanical or electrical engineering as well. And the focus there is to build upon your engineering knowledge, provide you with the necessary biological background, but also highlight how what are the nuances between the engineering knowledge and practice that you know when you are handling sensitive material like cells and proteins. And finally, we have a route for biochemical engineers, and that is really tailored to students with a biotechnology or biochemical engineering background. So could we have a quick shout out in chat what people's undergraduate degrees are? So can you just type your undergraduate degree? Okay, so we have chemistry. What else? Right, so it sounds like for, let me see. Oh, we have a couple more people online, so might as well wait for another couple of answers. So it sounds to me like the most relevant would be the route for scientists. That would cover most of you. So I'll go in detail over that, and then I'll maybe cover a bit more briefly the other two routes, just so I don't take up too much of your time. This applies to all routes irrespectively. So our entry requirement is a higher upper second class degree or the international equivalent, proof of English language, and these are example scores of what you would need to achieve. Your transcripts translated and provided in English if they're not already in English, a personal statement explaining why you want to study biochemical engineering, and two references from people who are able to comment on your suitability for the program. If you're still in uni, we need both of those references to be academic. If you have graduated and are currently working, we require at least one of those references to be academic. So the deadline for applications for this year is the 31st of July. However, particularly if you want to apply for a visa in order to study in the UK, I would urge you to apply as soon as possible, because particularly with all the thing with the coronavirus thing, the crisis, I'm expecting that processing visa applications at various places around the world will be substantially slower than they normally would be. So you want to make sure that you have your conditional offer, you have applied, you have received your conditional offer from us, and you can apply for your visa in time. Okay, so the MSc Biochemical Engineering for Scientists has 4 compulsory modules in term 1 and 3 compulsory modules in term 2. So term 1 is really fundamental engineering, so fluid flow, bioprocess synthesis and process mapping. This essentially teaches you about all the various pieces of equipment, or as we call them, unit operations, and what each one does. So you're probably all familiar with the concept of a reactor, but there's a lot of other equivalent pieces of equipment that are involved in the manufacturing of any biological product. We then have integrated downstream processing. So this is really all about, we take the material that comes out of the reactor and we now have to purify our substance out of that. Reactor engineering. And then in term 2, you'll do heat and mass transfer, bioprocess validation and quality control, and our biochemical engineering laboratory, which we have revamped. This year was the first year we ran the new version of this module. And the way we revamped it is we, instead of having, 2 hours a week, then you have to write a small report and so on, we said, let's take all of the learning outcomes and try to create a week-long campaign that would simulate as best we can uh, the actual process of manufacturing a batch of a particular product, right? Because products are manufactured in batches, and when you start this batch, usually the industrial term is that they start a campaign. So that will start by sterilizing all your equipment, inoculating your reactor, emptying the reactor, going through centrifugation, filtration, chromatography, all the way down to formulation. So we've tried to replicate this in a week-long activity. And finally, you will undertake a dissertation on bioprocess design in the summer months. So this in many engineering disciplines is known as a capstone project. It's a very important part of your professional development, and one of the necessary requirements in order for you to be granted chartered engineer status. And effectively, this is a group project. You work in groups of 4 or 5, and you're given a brief that you need to manufacture drug substance A using cell type B in country C. So you have to start by doing a market research. What is the demand in that part of the world? How much of the market can you capture? Based on that, you need to size your reactor. Based on sizing the reactor, you need to do excuse me, figure out how your downstream equipment will look like, and so on and so forth. And then in term 2, you also have an option between these 3 modules. So commercialization of research ideas, sustainable industrial bioprocess and biorefineries, and advanced safety and loss prevention. And I'll briefly go over the other 2 routes. They're probably, from what I'm guessing, they're probably not relevant to anyone currently attending, unless, let me see, chat might have been updated. Let me just check. No updates here. Term 1 is, sorry, I skipped a slide, is downstream processing, essentially biology 101, bioreactor engineering. Term 2 is biorefineries, bioprocess systems engineering, and the labs. And then in the summer months, they do a research project. And then they have some optional modules to choose from, 1 in each term. And the biochemical engineering route, I'll skip over to keep things short, because I don't think anyone based on the chat is from a biochemical engineering background. So I'll just wrap up what are the unique opportunities from the MSc Biochemical Engineering. It's your participation in this MBI program, which is the Modular Training for Bioprocess Industries program I talked about a bit before. Excuse me. And that is really a set of 4 or 5 day intensive workshops that have been designed for industrialists. And we invite academics from all around the world, leading academics from all around the world, and senior industrialists to present during these MBI sessions. So you really get not only the cutting edge, really latest developments in a particular research area, but you also get to hear from industrial experts and academic experts from all around the world. And on top of being able to start building your network by interacting with delegates from industry that are attending these events. The laboratory practicals that are uniquely structured because we have the pilot plant facility in our department. Unfortunately, this is a virtual open day and I can't go, but we do have a YouTube video that takes you around the pilot plant. So feel free to watch that on our YouTube channel. And it's really our links to industry and the environment that we foster that allows you to start building your professional network. In terms of graduate destination, the majority of our graduates move on to further studies. Around a quarter of our graduates move on to further studies, which shows the research intensive nature of our discipline. Around more than half end up in graduate jobs, mainly in research and development or manufacturing. A small fraction ends up in consultancy, and an equally small fraction ends up in the finance sector of London. Um, the average graduate starting salary figure shown here is just a ballpark figure. These things vary quite a bit depending on, social and political and financial stability. But that's a ballpark figure of what to expect. So with that, I'd like to thank you for your time. It's been a bit longer than usual, but we had to discuss a bit about the coronavirus thing. And I'm happy to open the chat now for any questions, and we'll probably welcome Carmen on the line, who's a student currently um, doing her MSc Biochemical Engineering, and she's doing a research project as well.
Carmen, perhaps you'd like to introduce yourself and just talk about what your research project is, just so we can get to know you.
Yeah, hi, hello everyone. So I'm Carmen. I did my undergraduate in biomedical engineering, and now I'm doing the engineering kind of part of the master that Alex was talking about. And I'm actually doing my dissertation with Alex. We were doing microalgae growth with the photobioreactor, and now we're kind of switching a little bit towards the modeling part of it, which I'm really excited about.
Okay, so let me start working through some of the questions already in chat. So the COVID, are we involved in it? I think I answered that. The answer is indirectly yes, in the sense that we've diverted a lot of our funding to support the efforts at Oxford. And we expect to be further involved once the substance has been identified, either a vaccine. I think Oxford is working on a vaccine specifically, so once the vaccine has been identified. Antibodies, are they extracted from plasma? It depends. It really depends. So there's no single answer to this question. And the antibodies are actually the ones that are being worked on more actively are for detection of the coronavirus rather than for treating the coronavirus. What else can be made through the biorefinery concept? All sorts of things, really. I've heard people building biorefineries around orange peel waste or municipal waste, or sugar is actually a very good example of biorefining. So you could take up the leftovers from the sugar canes that the sugar production industry would not use, and using specified enzymes, you can break those down into substances that cells can uptake, and you can start making ethanol, bioethanol out of that. Coffee beans is another thing that's or ground coffee, sorry, that is thrown away from all the coffee shops around the world, is something that people are trying to biorefine as well. So really, people's imagination is really the limit in terms of biorefining. And what was the other question? Do we collaborate with the FDA and NHS? We do collaborate with the NHS. I'm not aware of any direct projects we have with the FDA directly, or the NIH. Okay, other questions and questions for Carmen? Anything goes.
Just a quick one, which often comes up, is, Carmen, if you could just let us know what degree you did, where you did it, your undergraduate, and then just give us a quick idea what it was like coming to UCL Biochemical Engineering.
Okay, so yeah, I did my biomedical engineering undergrad in Purdue University, which is obviously a 4-year program. And I felt like over there we did way more theory than we are doing now at UCL. So coming into the biochemical engineering, I really like the small group, especially within the engineers. There's 7 or 8 of us that we're basically really close, and we help each other so much, like with the learning, because we all come from different backgrounds. I also really enjoyed the pilot plant week and the ultra scale down week, which were the laboratory course that Alex was talking about. In that one, you get a lot of knowledge of what you're actually going to be working on, which I believe from my undergraduate, we never had that overview of what would be the whole process. You would just focus on one small part of it in each different class. So these labs, I'm really happy that they put them all together in 1 week, because I believe you learn so much and you're so close with your group. You learn so much about communication skills because everyone's from all over the world. And yeah, it's just really great.
And you see, I'm glad you enjoy it, Carmen, because we put a lot of effort into revamping. But one of the things I'd like to point out, you see and you work with actual real life sized equipment. It's not a toy
Yeah.
reactor.
Mm-hmm.
Well, you will work on the small reactor on your bench, but you will see, and we will operate the actual large scale reactor as well. So you see
Yeah.
what a large scale centrifuge, what a large scale reactor. You see all of that equipment up close.
Mm-hmm. And then also with the MBIs, you get so much contact. Like I talked to so many people in Lonza that then once I went to the assessment center, I actually like, when I talked to the assessors about who I met from the company, they were like, oh my God, yeah, like we know them. I felt like that's really good networking for a person that's just like wanting to get into industry as soon as they finish the master's.
So a couple of questions here. So Stefan, that is an annual salary. That's not a monthly salary. And then the person that's on their mobile, I don't know the name. Is this a fully funded program? No, this is not a funded program. There are some scholarships available depending on your country. And if you go onto the FAQ section of our website, we have a link to a UCL portal that lists all possible scholarships, and you can filter by degree that you want to study, country of origin, and so on and so forth. But no, there are no department-based scholarships on offer.
Okay, we're only just over, so we do have time for a couple of minutes. I've checked and nobody else is queuing, waiting to get into our Zoom account, because we have to share it amongst all the departments in engineering. But if anybody's got any more questions for Alex or from Carmen, please pop them in either the Q&A section or the chat section. And as I said, if there's anything that occurs to you afterwards, by all means email myself or email Alex. We're more than happy to deal with it afterwards. Yeah, so thank you very much for attending. Thank you, Carmen, for joining. Thank you, Alex. And we'll just leave it a minute more in case any more questions come in.
Yeah, we can wait a couple of minutes for a few more questions.
Sure.
Usually, the more questions that are asked, the more questions come up. So it's good to wait to build that critical mass of questions. But as Kim said, if anything comes to you at a later point in time, just feel free to send me or Kim an email. And sorry for me coughing. I don't know what happened. My mouth dried up really. Okay. It seems we don't have a lot of questions. In which case, thank you all very much for attending. Thank you, Carmen, for taking the time to join in. And thank you, Kim, for hosting. Any questions, feel free to send us an email.